Jeff, can you see that?
Yep, it's showing. It's perfect.
And what should got it on the recording, yeah?
Yeah, I've recorded it. It's recorded.
Yeah, great. And can everyone see can you see the cursor?
Yeah.
The arrow? Hello?
Yeah, it's fine. Yeah.
You can see the arrow, yeah?
Yeah.
Great. Thanks, Jeff. Huge honor to be asked to give the Zangwill seminar in Cambridge. It's a particular honor to be asked to do it more than once. So I feel very honored and privileged to be able to speak to you today. Just a shame that we can't do it in person. So I was inspired to go back and look at what I talked about the last times I've talked to you in the department. So in 2008, I gave a talk called a role for hippocampal LTP in memory. It's not all associative. And then in 2014, is LTP really the basis of hippocampal long-term spatial memory? So you can see a bit of a pattern developing here. And I've come to the sad realization that I've only really got one talk. So a bit like Bill Murray in Groundhog Day, for those of you of a particular generation, I feel I'm destined to walk the earth giving the same talk over and over again. And far be it from me to break with tradition, but I'm going to talk about LTP again today. And as I'm sure most of you know, LTP is an experimental model of synaptic plasticity. It was first reported by Bliss and Lomo in the early 1970s. So they were performing experiments in anesthetized rabbits, and they were stimulating the perforant path input to the hippocampus and recording from the granule cell there in the dentate gyrus. And what they found was that if they gave a high frequency tetanus at the black arrows here, when they went back to giving single test pulses of a given size, they found that the field potentials had got bigger, suggesting that the synaptic connections between neurons had got stronger as a result of the high frequency stimulation. And ever since LTP was first reported, it's been considered a really important potential substrate for learning and memory. And we know an awful lot about LTP, particularly from experiments that have been done in hippocampal slices over the last 40 years. And we know, for example, that the most commonly studied form of LTP depends for its induction on the NMDA receptor subtype of glutamate receptor. So AP5 is a drug which blocks NMDA receptors, and you can see that it produces a very substantial block of LTP. The NMDA receptors, when they're activated, let a calcium signal into the cell, and that calcium signal acts as a trigger to initiate a whole series of different changes that go about to make the synaptic connection between the neurons stronger. And that, at least in part, appears to involve the trafficking or translocation of more AMPA receptors into the postsynaptic membrane. So these could either come from vesicular stores in the postsynaptic neuron, or more likely people think they're now coming from extrasynaptic sites out here on the edges of the synaptic zones. And so the idea is that you end up with more glutamate receptors on your postsynaptic membrane, and that should make the synaptic connection more efficacious or stronger. And this rapid trafficking of AMPA receptors into the postsynaptic membrane, at least in part, depends on the GluA1 subunit of the AMPA receptor. Now, a quick apology for terminology. So the terminology can get a little bit confusing, and that's partly because over the years, people have called this subunit different things, okay? So GluA1, GluRA, and GluR1 are all the same AMPA receptor subunit. Now, on some of the slides, it might be labeled as different things, but GluA1, GluR1, and GluRA are all the same subunit, and they're encoded by the gene GRIA1. And much of the talk today will be about this subunit, okay? So you need this AMPA receptor subunit to rapidly traffic more AMPA receptors into the postsynaptic membrane. So for example, if you take genetically modified mice that lack the GluA1 subunit, and you take hippocampal slices from those animals and try and induce LTP with a high frequency tetanus, you can see that the level of LTP is greatly diminished compared to what you see in wild type mice. So LTP depends on NMDA receptors for its induction, and at least in part, it depends on these GluRA or GluA1 containing AMPA receptors for its continued expression. Okay, so why do we care about hippocampal LTP? Well, obviously, it's interesting in its own right to try and understand the neurobiological substrates of learning and memory, but actually it's much more than that. And in the context of neuropsychiatry, deficits in hippocampal LTP have been associated with Alzheimer's disease, depression, and schizophrenia. So you can see it's potentially a very major player in some of the biggest neuropsychiatric disorders that we face. So for example, in Alzheimer's disease, an illness that I'm sure many of you are familiar with in your own lives, as well as in your scientific research, Alzheimer's disease often characterized, at least initially in the early stages, by episodic memory impairments. And we know, for example, that beta amyloid, protein that's very heavily implicated in Alzheimer's disease, suppresses LTP in hippocampal slices. These are experiments that were taken from by Olivier Shipton in Pauli Poulsen's lab when he was in Oxford. I could show you literally tens of different studies from mouse models of Alzheimer's disease, particularly PD, APP overexpressing mice, where scientists have taken slices from these mice and shown deficits in hippocampal LTP. And there's even evidence for reduced LTP in Alzheimer's patients, albeit from motor cortex rather than hippocampus. So lots of evidence linking deficits in hippocampal LTP with Alzheimer's disease. In terms of depression, there's also a big literature looking at the effects of stress and stress hormones like corticosterone on hippocampal LTP. So stress and corticosterone diminish LTP. This is just one example. This is a study using what's arguably the most widely used model of depression in animals to study for studying depression. So it's a chronic unpredictable stress model, so that the animals are exposed to lots of unpredictable stresses like immobilization, swim stress, overcrowding. They do things like tilting the cages, making the bedding wet, vibrating the cages, lots of things that just basically piss the animals off. And when you take slices from these animals at the end, you can see a quite dramatic reduction in LTP in the animals that have undergone the chronic unpredictable stress. So lots of evidence that stress and potentially depression might be associated with reduced LTP. And conversely, there's a growing body of evidence suggesting that antidepressant drugs like SSRIs might work by boosting plasticity processes. Now, this could include things like neurogenesis and structural changes, but it's also thought to include increases in synaptic plasticity as well. And these plastic changes might be one of the reasons why these drugs take several weeks to have a beneficial effect on mood in patients. Schizophrenia also seems to be a disorder of reduced synaptic plasticity. So over the last 20 years or so, there's been a tremendous amount of effort put into trying to establish the genetics of schizophrenia. And in the last 10 years, there have been some very high profile genome-wide association studies looking at the genetics of the disorder. And this paper in 2014 published a list of at the time 108 genes, which were considered to be the key risk genes for developing schizophrenia. I think that number is actually slightly higher now. But within these 108 genes, what scientists found was that there was an overrepresentation of genes associated with glutamate, with synapses, and with synaptic plasticity. And this included genes included genes for certain NMDA receptor subunits, and also the GluA1 gene for the GluA1 AMPA receptor subunit. And as I say, overall, there was an overrepresentation of genes in this list of 108 genes that were associated with synaptic plasticity, leading people to suggest that maybe we should consider schizophrenia as a disorder of reduced synaptic plasticity. Okay, so hippocampal LTP appears to be important in Alzheimer's disease, depression, and also in schizophrenia. And that leads to an immediate question, well, how on earth can LTP be doing all of these things? Now, it's true that there is overlap between the symptoms across these different disorders. So, for example, it's not uncommon to see psychosis and low mood in Alzheimer's disease. Likewise, one often sees cognitive impairment in depression and occasionally psychosis. And of course, there are cognitive and negative symptoms in schizophrenia. So there's certainly overlap across the different symptom domains in these three disorders. But at the same time, it's also true that episodic memory impairments are clearly not the same thing as low mood and are clearly not the same thing as psychosis. So how on earth can hippocampal LTP be doing all of these things? And in the case of schizophrenia in particular, there's a further paradox, which is if schizophrenia is a disorder of reduced synaptic plasticity, then how does that fit with the textbook view that LTP is the basis for associative memory formation? Because arguably, and many psychiatrists would say that schizophrenic patients are actually more likely to form associations rather than less. And it's these maladaptive or false associations that lead to psychosis and delusions. And it's not clear how then a disorder of reduced plasticity would lead to an increased likelihood of forming maladaptive or aberrant associations. So people have been interested in hippocampal LTP and these disorders for many years now, and the scientific community, both academia and industry, have spent enormous sums of money targeting hippocampal LTP as a potential way of developing new treatments for these different disorders. And I think it's fair to say that this approach has so far proved fairly unsuccessful, if completely unsuccessful, if not completely unsuccessful. And what I want to argue today is that lack of success, or that failure, at least in part reflects the fact that we don't really understand exactly what the cognitive and psychological sequelae of hippocampal LTP actually are. And in particular, I'm going to argue that the traditional textbook view that hippocampal LTP is important for forming excitatory associations between stimuli. So for example, learning to associate a particular location in the water maze with the presence of an escape platform is actually incorrect. And actually we need to find a different way of thinking about what LTP is doing in the hippocampus. So before I show you any data, I just want to take 5 minutes or so to reevaluate the hippocampal LTP memory hypothesis and just touch on a few of the issues that have remained in the field without a really satisfactory explanation. The first of those is the time course of learning-induced LTP. Now, it's certainly true that if you induce LTP with a high-frequency tetanus, you can get an increase in synaptic strength, which lasts for weeks, if not months. So in a follow-up paper that Tim Bliss published with Gardner-Medwin around the same time as Bliss and Lomo, they reported that if they used chronically implanted electrodes and applied a high-frequency tetanus, they could still see LTP several weeks after induction. And subsequently, other groups have replicated this phenomenon. And I think Cliff Abraham in New Zealand actually at one stage had evidence of LTP, electrically induced LTP, that lasted for at least a year. So certainly artificially induced LTP with a non-physiological high-frequency tetanus can be very long-lasting. But actually, when we look at LTP that's induced as a consequence of learning, there's actually quite a different result. And actually, these experiments where you try and demonstrate LTP as a result of a behavioral experience or a learning episode have actually proved really difficult to do. And there aren't many very good studies that have managed to do this. One of the most widely cited is this paper from Whitlock and colleagues from Mark Baer's lab. And they trained rats on an inhibitory avoidance task, so a simple learning task, and then looked to see whether there were LTP-like changes that accompanied the learning in these animals. And they did that using electrophysiological methods, but they also used an alternative approach, which was to measure the levels of the GluA1 subunit in the postsynaptic density of the neurons. Okay, so you remember LTP, at least in part, involves translocating or trafficking more GluA1 containing AMPA receptors into the postsynaptic membrane. And the idea is that if you can measure the levels of GluA1 in postsynaptic membranes, you've got a marker of how much LTP there is. And the argument being that if learning is a result of LTP, you should see an increase in GluA1 levels in the postsynaptic densities. So they trained the animals up, took brains, isolated postsynaptic densities, and measured levels of GluA1 or GluR1. And you can see that there's an increase in GluR1, there's also an increase in GluR2, and there's also an increase in the phosphorylation of GluR1. So very clear demonstration of a change in AMPA receptor levels in postsynaptic density as a result of the behavioral experience. But what's striking is that in actual fact, these changes last for about 2 hours. They certainly don't last as long as the memory trace. If you look at the avoidance response in these animals, it's still very strong at 24 hours. And I'm sure if they tested the animals after several weeks, they would still have seen strong memory performance. Yet the memory trace in the hippocampus, at least as far as the AMPA receptor levels are concerned, is back down to baseline within a couple of hours. So clearly, there's a disconnect there between the memory trace in the hippocampus and the memory that's being expressed at a behavioral level. And this is potentially consistent with some other ideas that have emerged over the last couple of decades, which is the idea that there is a profound synaptic homeostasis that occurs during sleep. So this is an idea that was first put forward by Tononi and Chiarelli, and they argued that when we're awake, we experience lots of things, there's lots of LTP-like events, and so synaptic weights overall show an increase. But of course, you'd very soon saturate those synapses if that was all that ever happened. So there needs to be some way of downscaling or reducing those synaptic weights to avoid saturation, and also to potentially to avoid inefficient energy usage by the neurons. And so what they've argued is that during sleep, and in particular during slow wave sleep, there's a mass or fairly global synaptic downscaling, like an LTD-like event, which reduces the strength of the synapses when we're asleep. And evidence for that comes from this study by Vlad Vyazovsky, who's now a colleague of ours in Oxford. But when Vlad was working with Chiarelli and Tononi, he measured GluR1 or GluA1 levels in postsynaptic neurons in just the same way that Whitlock had done. And he showed that if you compared animals that had been asleep for 6 hours with animals that had been awake for 6 hours, you can see much higher levels of GluR1 in the animals that have been awake compared to the animals that have been asleep, suggesting that when we're awake, we get lots of LTP, synaptic weights get stronger. But then when we go to sleep, those synaptic weights undergo a downscaling or depotentiation or LTD, such that synaptic weights are reduced back down to their original level. And of course, one implication of this is that these memory traces that we're looking at are effectively being erased during slow wave sleep. So whatever's being represented in the hippocampus in terms of glutamate receptor changes is actually being wiped out as soon as we go to sleep. A third issue that the field hasn't really dealt with particularly well is the idea that there are multiple mechanisms of LTP, often summating at a single synapse. So up until now, I've been telling you that there's the most commonly studied form of LTP depends on NMDA receptors for its induction, and then trafficking of GluA1 containing AMPA receptors for its expression. But actually, it's possible to induce quite substantial levels of LTP in the GluA1 knockouts if you use a different kind of induction protocol. So this is a study from Carola Rombergs. Some of you might remember Carola. She worked with Tim and Lisa as a postdoc in Cambridge a few years back. But in her previous life, before she came to Cambridge, she did a PhD with Ole Poulsen when Ole was in Oxford. And Carola looked at LTP in wild type and GluA1 knockouts, but using a theta burst stimulation. So it's a different kind of LTP induction protocol for inducing LTP. And what you can see at the trace at the bottom is that there is now substantial LTP in the GluRA knockouts when you use a different kind of induction protocol. In another study from Roger Nichols' lab, they also showed that it's possible to get substantial LTP in a GluA1 knockout animal, and that was supported by the GluA2 subunit. They actually went on to show that it was possible to get LTP in the complete absence of AMPA receptors, and that the LTP was supported by kainate receptors. But the take-home message, and this, the possibility that there may be different forms of LTP is consistent with the idea that there are different trafficking mechanisms for AMPA receptors into postsynaptic densities. So there is trafficking involving GluR1 or GluA1 containing receptors, but then it's also possible to get trafficking of GluR2, GluR3 receptors as well, and that these two trafficking mechanisms can be dissociated. So there are different ways in which AMPA receptors can be trafficked into the postsynaptic density. And I guess a key question then is, what's the functional significance of having multiple LTP mechanisms at a single synapse, where these mechanisms appear to be additive and both act together to potentially increase the strength of synaptic connections? A final point just to bring up in terms of reevaluating the LTP memory hypothesis is the empirical data itself. And I've lost count of the number of studies that I've done over the years where we've looked at models, whether it's pharmacological or genetic, where there's impairments in hippocampal LTP, but in actual fact, the animals can learn the standard water maze task every bit as well as their controls. I'm going to show you just one example, which is a study from the Kandel Tonegawa supergroup. So they made a mouse in which NMDA receptors were knocked out from the hippocampus. And the claim was that these animals were unable to learn the Morris-Water maze. However, in the following years, it became very clear that the knockout wasn't specific to the hippocampus and extended widely into cortex. And when we revisited this with a knockout mouse made by Peter Seeberg and Rolf Sprengel in Heidelberg, so a mouse that lacks NMDA receptors in the hippocampus, lacks LTP in the CA1 subfield, we found that actually these mice could learn the standard water maze task perfectly well. So standard water maze task, there's a fixed platform location. The animal starts from different points around the edge of the pool, and the animals are required to always go to the same place to escape from the water. And you can see that the knockout mice learn just as well as the controls. On a probe test where the platform is removed and the animals are allowed to swim around freely, what you can see is that if anything, the knockouts do slightly better. So maybe the traditional textbook view that LTP in the hippocampus is important for forming associations, for example, between a spatial location and the platform, is potentially misguided. Okay, so what is LTP doing? Well, when we think about learning and memory, we often think about encoding and consolidation and retrieval, and then sometimes also reconsolidation. But there's this other thing called priming, okay? And ideas about priming have been around for a long, long time. This is a quote from William James in the late 19th century. Nothing is easier than to imagine how when a current once has traversed a path, it should traverse it more readily still a second time. Sounds a bit like LTP. Okay, this is a definition of priming taken from an undergraduate psychology textbook from Gazzaniga. Priming refers to a change in the response to a stimulus as a result of prior exposure to that stimulus. Forms of objects or words that have been presented previously or primed are more easily recognized. We are faster at identifying them than our forms that we have not previously been presented to, i.e. not primed, okay? And another way we can think about priming is that priming is something that reduces surprise. So if you're thinking about something already, it's going to be less surprising. Something surprising if you haven't been thinking about it, okay? And we know from many years of psychology research that there are two basic forms of priming. There is self-generated priming. So this reflects a stimulus that's present in the real external world. So it generates a sensory memory. It's something that you've experienced in the real world. If you've experienced something recently, you're going to be less surprised if that stimulus is presented again. So self-generated priming reduces surprise because you've experienced that stimulus recently. Things can be less surprising because of what you've learned in the past. So they can be less surprising because what you retrieve from long-term memory leads you to predict that something might happen. So the second form of priming is retrieval-generated priming, and that's based on long-term associations and associative recall of memories, such that if you've thought about something recently and then it happens again, it's going to be less surprising. And there are some very elegant psychological models that integrate ideas of priming with attention and memory. The most popular is this sometimes opponent process model. It was described by Wagner in the late 1970s and early 1980s. So Wagner conceptualized stimulus representations as existing in one of three states: an inactive state, an A1 state, which is like the focus or the forefront of attention, and an A2 state, which is like a secondary or refractory state, which reflects priming. So when things are in the A2 state, they are less surprising, and things can get to the A2 state from the outside world through self-generated priming, or they can get to the A2 state from long-term memory through retrieval-generated priming. And there are some important extensions of this model that followed on, including the MSOP model, which was developed in your department in Cambridge, and then also Wagner's affective extension of SOP, which we'll touch on towards the end of the talk today, if there's time. Okay, so how might priming help us think about the role of hippocampal LTP in schizophrenia, for example? So just a quick recap: schizophrenia, we've said, is now being considered as a disorder of reduced synaptic plasticity, and genome-wide association studies have implicated a number of genes associated with plasticity, including GluA1, the gene that codes for this GluA1 AMPA receptor subunit. Now, importantly, it's not just genetics that links GluA1 to schizophrenia. There's also a considerable amount of post-mortem data from patients, much of which was conducted by a colleague here in Oxford, Paul Harrison, showing reduced levels of GluA1 in the brains of schizophrenic patients. And importantly, Paul did a number of studies to show that that wasn't a consequence of long-term treatment with antipsychotics. It was a genuine effect of the illness rather than the medication. So lots of evidence linking this GluA1 AMPA receptor subunit to schizophrenia. And first thing to say about GluA1 is that if you knock it out in a mouse, the mice can learn the standard water maze task perfectly well. So they're perfectly capable of forming associations, for example, between a particular place and the presence of a platform, despite there being an impairment in hippocampal LTP. And to cut a very long story short and condense 20 years of research into one sentence, we now believe that GluA1 and GluA1-dependent plasticity is important for this self-generated priming, making things less surprising because they've occurred recently in the real external world. And deficits in self-generated priming result in impairments in short-term habituation. Okay, so if priming reduces surprise, if priming is impaired, then things will stay surprising and attention-grabbing for longer. And as I'm sure you all know, habituation describes the decline in the tendency to pay attention to stimuli that have become familiar due to prior exposure. Okay, so I'm going to give you a quick example of this by getting you to play a little game. So we're going to play Spot the Difference. Okay, you all ready? You remember him? Yeah. Does anyone remember a band in the early 80s called Flock of Seagulls? This is what happens when Flock of Seagulls get old. Okay, so everybody spend some time looking at Donald. Okay, you all had a good look at Donald? Okay, now hopefully when Boris appeared, everyone oriented and looked at Boris. Okay, now that's not because Boris is more handsome than Donald or because Boris is any less dangerous than Donald. It's just that you'd already spent some time attending to Donald. You knew about Donald. There was no need to keep looking at Donald. And so when a novel stimulus appears, you'll pay more attention to the novel stimulus. That's because you've habituated to Donald. Now mice don't care about Boris and Donald, but we can use tasks like the classic object recognition task to look at these short-term habituation processes. So for example, we can expose the mouse to two copies of the I Love Tea mug, and then after a short interval, we can give them an I Love Tea mug and a Carlsberg beer bottle. And what you'd expect is that a normal animal would spend more time looking at the beer bottle than looking at the I Love Tea mug, because they've habituated already to the I Love Tea mug. They don't need to spend more time paying attention to this because they already know about it. Now, of course, Carlsberg don't do behavioral neuroscience, but if they did, the data would look like this. You get a nice novelty response in the wild types, but in the GluA1 knockouts, they're at chance. So they fail to recognize the familiar stimulus as familiar, and they keep exploring both stimuli to a high degree and show no preference. Okay, we can do the same thing in the spatial domain using a simple spatial novelty Y maze task. So in this experiment, the animals are allowed to explore two arms of a Y maze. So it's a perspex, transparent Y maze with lots of spatial cues around the room. The animals are allowed to explore two arms of the maze, and then after a short delay, they're placed back in the maze, and now they're allowed to explore all three arms. And of course, what you'd expect is that the wild types have habituated to these two arms, so they would show a strong preference to the novel arm over the other arm and the start arm. And that's exactly what we see. Wild types show a strong preference to the novel arm. But what's striking is that the GluRA or GluA1 knockouts show no preference at all. And what's striking is that they're as bad as hippocampal lesioned animals here on this novelty preference test, where if you remember, they were as good as wild type controls in the water maze. So this isn't a general problem with spatial processing. It's a very specific deficit in short-term habituation. And actually, we can mess around with the parameters of the task. And when we do that, we see something even more interesting. So if you've fallen asleep in the last 10 minutes, now's the time to wake up, because now the data is going to get really interesting. Okay, so we're going to do a similar experiment, but now we're going to place the animals in these little open fields that have got distinct wallpapers and floor inserts. So we can generate two different contexts, A and B. And all we're going to do is we're going to place the animals in for 5 minutes at a time with 1 minute gaps. And we're going to look at how much the animals run around and explore the boxes. So we're going to look at their locomotor activity levels, and we're going to use that as a proxy of how much attention they're paying to the environmental cues. Okay. Now, half the animals are going to go through box A, box A, box A, and half the animals are going to go through box A, and then on the third trial, they're going to be exposed to a different box B, okay? And of course, what we'd expect is that normal animals will habituate and explore less and less across the 3 exposures to A, but then exploration will be reinvigorated when the animals are placed in B. And that's exactly what we see in the wild type mice. So their levels of exploration go progressively down across the 3 trials. When we replace context A with context B on trial 3 here, in these animals, you can see that the levels of exploration go back up, demonstrating that this habituation is stimulus specific. It's based on a memory of stimulus A, and it's not just a general change in arousal or fatigue, okay? So what about the GluA1 knockouts? Well, strikingly, they do exactly the opposite. So they start off running around the box, quite similar to the wild types on trial 1, but rather than go down, their levels of exploration actually increase from trial 1 to trial 2. So their levels of exploratory attention are actually increasing with each exposure rather than going down. When we change the stimulus on trial 3, you can see that levels of exploration go back down again. So this sensitization, this increase in attention that the GluA1 knockouts are paying, again, is stimulus specific. It's present based on recent experience of A, but if you go to a novel stimulus, levels of attention go back down again. And this is a really important result because it shows that the GluA1 knockouts do have a memory of context A, but they're expressing that memory in a very different way from the wild type mice. We've done something similar with MK801. So MK801 is an NMDA receptor antagonist. Again, nice stimulus specific habituation in the saline treated animals. But again, animals with an NMDA receptor blocker, so presumably LTP is blocked in these animals as well. You can see that the levels of exploratory attention are actually increasing across the trials. And again, that sensitization is stimulus specific. So whether it's GluA1 knockout or pharmacological blockade of NMDA receptors, the levels of attention that are being paid to cues in the environment are actually increasing rather than decreasing when we block LTP in the hippocampus. We've done something similar with lights in an operant box. So in this experiment, we just measured how much animals looked at different light cues on the wall of the operant box. And again, what we did was we presented them with a light, and then 30 seconds later, we either presented the same light again, or we presented a different light. And what we found was that in normal wild type mice, orienting goes down, reflecting habituation. So the first time they see the light, they go, oh, that's a nice light, that's interesting. You show them the same light again, and orienting goes down. Yep, not interested in that light, seen it already. Again, what's striking is that the GluA1 knockouts do the opposite. For the first time they see the light, they go, oh, yep, that's a light, that's nice. Second time they see the light, they go, wow, that is the most amazing light I've ever seen in my life. And they pay much more attention to the light the second time they see it compared to the first, and much more than they do, again, to a completely novel light. So the levels of attention that these GluA1 knockout mice are paying to this light cue is actually increasing with exposure rather than decreasing. And this is very similar to something called aberrant salience, which if any of you work on schizophrenia in patients, you'll know is a widely used endophenotype in schizophrenia research, and it reflects the maladaptive, increased and aberrant levels of attention that are paid to stimuli in patients. And just to give you one example of that, this is anecdotal reports taken from a paper by Kshitish Kapoor. Anecdotal reports taken from the prodrome of the disorder. So this is the phase of schizophrenia before they're diagnosed formally with the disease. So patients talk about having developed a greater awareness of things. My senses were sharpened. I became fascinated by the little insignificant things around me. Sights and sounds possessed a keenness. My senses seemed alive. I'd noticed things I'd noticed things that I'd never noticed before. My capacities for heightened sensory receptiveness was very keen at that time. I'd had the same intensity of experience at other times when I was normal, but such periods were not sustained for long. So hopefully from that, you can get this sense that these individuals are showing a sort of heightened perceptual awareness of things in their environment. They're paying more attention to cues in their world, just like the GluA1 knockout mice with the lights on the wall of the operant box. And aberrant salience is classically associated with a hyperdopaminergic state, increased levels of dopamine, particularly in the striatum. And so over the last few years, in collaboration with my close colleague Mark Walton in Oxford, we've tried to get a way of measuring dopamine levels in these GluA1 knockout mice. And to do that, we've used an approach called fast scan cyclic voltammetry, which is a voltammetric approach which gives you a real-time readout phasic dopamine signals in the brains of behaving animals. And this purple blob here is dopamine. And this, interestingly, this is the dopamine response to a neutral light cue. So there's obviously a huge literature talking about dopamine and reward prediction errors, but actually you can see very robust and reliable signals to any surprising or unsignaled stimulus in the world. So this was an unsignaled light cue, and you can see a very strong dopamine response. Okay, so we've looked at dopamine levels in these GluA1 knockouts recently. And the first thing we did was just look at responses to electrical stimulation of the VTA in anesthetized animals. So we had an FCV recording electrode in nucleus accumbens, stimulating electrode in the VTA, and we compared the size of the dopamine signals in wild types and knockouts. And what you can see is that across a range of stimulation intensities and stimulus pulse numbers, that actually the two genotypes are very similar. There's no differences between the knockouts and the wild types, suggesting that the intrinsic properties of the dopamine neurons are actually pretty normal in the GluA1 knockouts. Okay, so what happens when we look at dopamine in behaving animals? Well, as we've suggested, obviously dopamine neurons care about rewards. So the first thing we did was look at unsignaled rewards in GluA1 knockout and wild type animals. And lo and behold, what we found was that the dopamine signals were much bigger in the knockouts than in the wild types. So this is the average dopamine signal for unsignaled rewards in the knockouts and the wild types. Now, this is the signal. These are the averages taken across the whole session. So the sessions were about 40 minutes long. This is the average across the whole session. When we looked trial by trial, what we found was that actually, to begin with, the signals were fairly well matched, but you can see a quite rapid habituation of the dopamine signal in the wild types, whereas in the knockouts, the signal remains high for most of the session. So maybe this hyperdopaminergic state reflects these deficits in short-term habituation. So we did the same thing with light cues. So again, we measured the phasic dopamine responses in accumbens to unsignaled light cues. And again, what we found was that the signals were much bigger in the knockouts than in the wild types. So an LED light stimulus. Now, again, this is the average taken across a whole session. What we did was we split the session into first and second half, so early and late, and compared the dopamine signals in the two genotypes across the two halves of the session. And what we found was that in wild types, again, the dopamine signal habituates across the session. So you get a big dopamine response early in the session, but that signal reduces in the second half of the session. Whereas in contrast, in the knockouts, there is no habituation to the light stimulus across the session. And so the deficit in habituation could potentially explain that hyperdopaminergic state in the GluA1 knockouts. Again, we can look at same and different cues. So again, the prediction would be that if you present the same stimulus twice, you'll get a smaller dopamine response compared to if you present two different light cues. And that's exactly what we saw in the wild type. So there was a smaller response to the same than the different cue. Again, that same different effect is missing in the knockouts, consistent with the fact that deficits in short-term habituation are driving this hyperdopaminergic state. Okay, so that's quite nice. That's evidence that a core attentional deficit, so impaired short-term habituation, resulting from an impairment in glutamatergic transmission and deficits in synaptic plasticity, could be a driver of a hyperdopaminergic state leading to psychosis in schizophrenia, potentially providing a link between the glutamate hypothesis of schizophrenia and the dopamine hypothesis. Now, obviously, the follow-on implication of that is that if you're paying inappropriate and elevated levels of attention to the wrong cue at the wrong time, and you have inappropriate phasic dopamine signals to these cues, then that could lead to the formation of false associations and false beliefs. And this could be the driver of the maladaptive beliefs that underlie delusions in psychosis. And in a recent series of experiments by Marios Panai and Tom Blackmore in the lab, we've started to look at this using a simple sensory preconditioning paradigm. So in this experiment, what we did was we had a pre-exposure phase, a conditioning phase, and a test phase. In the pre-exposure phase, animals are exposed to pairs of auditory cues. Then during conditioning, one of the cues is paired with a food reward. And then in the test phase, we ask them, how do you feel about QA compared to QC? And what we would expect is that a normal animal would respond a lot more to A than it will to C, because it's learned to associate A with B with reward. Therefore, we'd expect more responding to A than to C. And what we found was that actually the sensory preconditioning effect in the wild types was pretty weak in this experiment. but it was very robust in the GluA1 knockout. So they responded a lot more to A than to C, and suggesting that the associations they formed between A and B were much stronger during the pre-exposure phase. Conditioning rates were similar in the two genotypes. So again, suggesting that they've learned this A-B association more robustly than the wild type mice, consistent with this idea that they might be more likely to form associations rather than less. Now, of course, you might turn around to me and say, well, that's not schizophrenia, that's cognitive enhancement. That's a good thing. Learning's better here. Okay, so what happens if we test these animals in a slightly more complex situation? So now we have pairs of Q. So QA is paired with B, QC is paired with D. Then they go through a conditioning phase where B is rewarded and D isn't. And then we ask them how they feel about A and C. And of course, what we would expect is that a normal animal will respond much more to A than it does to C. And that's exactly what we found. The wild types responded much more to A than to C. But interestingly, now the knockouts don't show any preference for A over C. And importantly, they respond to C exactly the same way they respond to A. So they respond to C as if they're expecting a reward in the same way that they respond to A. And so one possibility is that this increased likelihood or propensity to form associations means that they now start to form associations across the pairs of stimuli as well as within the pairs. Now, there may also be a contribution here from the conditioning phase. We think that actually it's a combination of what's happening in the pre-exposure phase and the conditioning phase here. But either way, what you end up with is a maladaptive association in the knockouts where they treat C as having a meaning that in actual fact it doesn't. So potentially consistent with the idea that deficits in short-term habituation change the way in which associations are formed, which could potentially lead to maladaptive associations and maladaptive beliefs. And as I said, this might be an explanation for how deficits in synaptic plasticity, which impair self-generated priming and impair short-term habituation, could lead to the formation of aberrant associations. An important question is where is this happening in the brain? So far we've been talking about GluA1 knockout mice, which are very old school. These are constitutive whole brain knockouts. So the GluA1 subunit is missing throughout the lifetime of the animal and throughout the entire central nervous system. And so in theory, the deficit could reflect an absence of GluA1 anywhere in the brain. And of course, if we think about priming and habituation, then it wouldn't be the greatest surprise if these were more general properties of a number of different brain networks. And so whereas I wouldn't want to say that it's all hippocampal, there's certainly some very good evidence that it's at least partly a reflection of GluA1 dysfunction in the hippocampus. And we've done a couple of experiments recently to try and get at this. One study that we've done, which we're writing up at the moment, is in collaboration with Radu Aricescu, who I think is now at the LMB here in Cambridge. We made an antibody that binds to the GluA1 subunit of the AMPA receptor, so we can use it a bit like a pharmacological antagonist to block GluA1 function in otherwise normal wild-type mice. And when we do this, what we find is that there's a robust and reversible deficit in short-term memory when we disrupt GluA1 function with the antibody. So that's nice. It says it's at least in part hippocampal, and also that it reflects the absence of GluA1 or a deficit in GluA1 function in adulthood and not just in development. In another study, which we published recently, Alexei Bygrave and Dennis Keitzel used a viral approach to reintroduce GluA1 back into the hippocampus of an otherwise GluA1 knockout mouse. So they took GluA1 knockouts and using a viral approach, they were able to reintroduce GluA1 into the hippocampus, and particularly into the CA3 subfield of the hippocampus. And what they found was that, so this is the Y-maze novelty preference task that we looked at before. This is the wild types. These are the GluA1 knockouts. Strikingly, when they put GluA1 back into the hippocampus, they produced a near complete rescue of short-term memory performance, short-term habituation in the otherwise GluA1 knockout mice, providing very nice evidence that at least in part, it reflects a role for hippocampal GluA1 and GluA1-dependent synaptic plasticity in these self-generated priming short-term habituation processes. Okay, so how are we doing? So going back to Wagner's model, if we block GluA1, we prevent self-generated priming, so we prevent the stimulus representation passing from the A1 state to the A2 state, suggesting that maybe GluA1-dependent LTP, at least in part in the hippocampus, is what underlies self-generated priming. Okay, so back to how can hippocampal LTP be doing all of these different things across the various psychiatric disorders? Well, obviously one possibility is that the different flavors of LTP, the LTP with different underlying mechanisms, may be important for supporting different aspects of priming. So we've suggested that there are two main forms of priming, self-generated and retrieval-generated, and hopefully I've convinced you that GluA1 is important for self-generated priming. We know that GluA1 isn't necessary for retrieval-generated priming. So for example, if we look at long-term habituation or object in context tasks where clearly the memory processes required involve retrieval-generated priming, the GluA1 knockouts are absolutely fine. In some cases, actually, long-term habituation looks enhanced in the GluA1 knockouts. So retrieval-generated priming doesn't require GluA1. Okay. So one interesting possibility is, well, maybe GluA1-independent forms of LTP could underlie retrieval-generated priming. So what do we know about GluA1-independent LTP? Well, if we go back to Carolla's work, we know that, for example, if you induce GluA1-independent LTP in a GluA1 knockout, you can block it with an NMDA receptor antagonist like CPP, suggesting that GluA1-independent LTP, again, at least in part, depends on NMDA receptors. Okay. What happens when we knock out NMDA receptors in the hippocampus? So I told you a little bit about this mouse earlier on in the talk. This is the mouse that was made in Heidelberg by Rolf Sprengel, and it provides a knockout of NMDA receptors in CA1 and dentate. It's not perfect. There's some expression in the olfactory bulb, but you can see across the rest of the cortical mantle, there's almost complete sparing. When we take slices from these animals, we get a deficit in LTP and CA1. And I showed you before that these mice can learn the standard version of the water maze task perfectly fine. But if we run the water maze in a different way, we get a very different result. So we took separate cohorts of experimentally naive animals, and we ran them on a different version of the Morris water maze. So now there are two black footballs sitting on the surface of the water. One sits on the escape platform and provides a beacon so that the animal knows where to go. And the second one is anchored to the bottom of the pool, so it acts as a decoy, but it provides no escape from the water. And these two black footballs are visually identical, okay? And so the only way that the animal can discriminate between the two of them is using the allocentric spatial cues around the room. And again, like in the standard water maze task, we vary where the animal starts from. And the idea is that they have to choose the correct black football defined by spatial cues in order to escape onto the platform. And we found that if we trained them up and then did the probe test where we take away both of the beacons and the platform and ask the animals, do you know where the platform is? Then the knockouts do just as well as the controls. Again, if anything, slightly better. So they know exactly where the platform is relative to the cues around the room. But what we also found was that if we scored their performance in a different way, so if we counted the number of errors that they made, so the number of times that they swam up to the wrong black football, then actually what we then found was a massive increase in errors in the hippocampal NMDA receptor knockouts. So they were much more likely to swim up to the wrong black football. So they made a lot more errors during training. And what we found was that these errors were particularly dependent on where the animals started in the pool. So we systematically varied where the animals started from. On a third of trials, they started equidistant. On a third of trials, they started close to the S+, so close to the correct black football. And on a third of trials, they started close to the S-. And what we found was that the errors, so when they swam to the wrong black football, were almost entirely on trials where they were starting close to the decoy. You can see that on S+ starts, they all do really well. When they start halfway between, there's a small reduction, but statistically, there's no difference between the two genotypes. Whereas on the S- start position, so when they start close to the decoy, both groups do really badly to start with, but then the knockouts are much slower to learn to inhibit that response to swim to the first black football that they see. As a control study, we ran a visual discrimination with two now visually distinct beacons, so a black and white cylinder and a gray funnel. Ran this task in exactly the same way, and now we found no impairment whatsoever, even on those S- start trials. So even when we started them close to the wrong beacon, now the knockouts were every bit as good as the controls at swimming to the correct beacon and getting out of the water. And so to summarize that, we've got an impairment on a spatial discrimination, no impairment on a non-spatial visual discrimination. And of course, it's tempting at that point to suggest, well, maybe that's because this is a spatial task and this is a non-spatial task. We know the hippocampus is important for space, place cells, et cetera, et cetera. But of course, we also know that the knockouts are perfectly capable of learning the standard spatial version of the water maze task. If anything, they end up doing slightly better than the controls. And so instead of this being about whether a task is spatial or not, we've argued that it reflects the ambiguity of the black football in this version of the task compared to the situation where you've got two completely distinct and orthogonal cues like the cylinder and the funnel. So in the spatial discrimination with the two black footballs, the black football generates two memories. There's a memory of football leads to escape, and there's a memory of football leads to no escape. And so maybe hippocampal NMDA receptors and hippocampal NMDA receptor dependent LTP is important for disambiguating or pattern separating between overlapping or competing memories. Okay, so how might priming help with the disambiguation between overlapping and competing memories? Well, we know a lot more about what might be happening at an electrophysiological level in terms of what during these disambiguation processes. And this stems from work, fantastic work that was done by Dave Reddish in the States in the early 2000s, and then has been followed up by groups like the Moses Group and also by my colleague in Oxford, David Dupre. And what Reddish showed was that if you look on a very fast timescale in the hippocampus, you see that even in a single spatial location, you get the instantiation of multiple cognitive maps at the same time. And for example, during a disambiguation process, you might have two cognitive maps, one cognitive map reflecting one memory and one cognitive map reflecting another memory. And when I talk about a cognitive map, these are cell assemblies of place cells in the hippocampus that are firing at a particular rate. So you might get one representation which reflects one memory and another representation that reflects another memory. And during a disambiguation process, what you find is that you get rapid activation of both maps across successive cycles of the theta wave. So you might get cognitive map 1 in the first theta cycle, then cognitive map 2 in the second, then back to 1, then back to 2, then back to 1, and then back to 2. And you get this rapid flickering between these different cognitive maps. And the idea is that this is reflecting the disambiguation process that's going on between the hippocampus and the rest of the brain, such that the hippocampus is gathering more information to decide which memory to retrieve and to act on. And of course, if this disambiguation process involves the rapid reactivation of memory traces, it wouldn't make sense if those memory traces had to be retrieved from scratch de novo each time. So being able to tag or activate or prime these memory representations would greatly facilitate the disambiguation process that might be going on in the hippocampus. So priming the memories and retrieval generated priming of the memories is going to really facilitate the disambiguation process that's going on when we have to retrieve memories accurately through the hippocampus. And of course, when it comes to Alzheimer's disease particularly associated, at least in the early stages, with episodic memory impairments. And so deficits in LTP, which might impair retrieval generated priming and might disrupt this disambiguation process, could be the driver of the episodic memory impairments in patients. How am I doing for time, Jeff?
Fine, another 5 minutes or so.
Yeah. Okay. So just very quickly, then I'm gonna finish very quickly. Just to talk very quickly about depression. This is much more speculative, and I've got much less data to show you here. But I just wanted to throw this out there as a possible interesting angle on how boosting plasticity and potentially synaptic plasticity in the hippocampus might be important for the action of SSRIs in treating depression and improving mood. So back to the hippocampus, we know that the hippocampus is actually more than it could be thought of as more than one structure, in that there are very profound differences between what the different subregions of the hippocampus are doing. So we might think about the hippocampus having a septal pole and a temporal pole. So there's a dorsal hippocampus up here and a ventral hippocampus down here. For those of you that are more familiar with the primate brain, the dorsal hippocampus would be the equivalent of the posterior hippocampus in primates. And the anatomical projections to and from dorsal and ventral subregions of the hippocampus are very different. So dorsal hippocampus receives highly processed sensory information through sensory cortices, association cortices, entorhinal, perirhinal cortex. Ventral hippocampus, on the other hand, is much more closely associated with the amygdala, for example, with structures in the HPA axis and with certain frontal regions that might be important in emotion. And what we know from lesion studies over many years, including work from our own lab, is that actually you can doubly dissociate different functions between the different hippocampal subregions. So dorsal hippocampal lesions reduce deficits on spatial memory tasks. Ventral hippocampal lesions reduce anxiety, but ventral lesions have very little effect in the water maze. Dorsal hippocampal lesions have no effect on the elevated plus maze. And as I say, this is potentially consistent with the anatomy whereby dorsal hippocampus is receiving highly processed sensory information. Ventral hippocampus is much more closely tied to areas involved in regulation of emotion, like the amygdala and the HPA axis. That said, it also seems likely that there will be some sort of common algorithm or function that's being performed by both the dorsal hippocampus and the ventral hippocampus across these different behavioral domains. And one interesting possibility is that this difference between sensory and emotional hippocampus, the sensory and emotional hippocampus, might map onto ideas that Wagner went on to suggest in later iterations of his model, when he suggested that memory representations have both a sensory and an emotional component. So there are sensory elements and emotional elements, and that these are processed on slightly different timescales, but according to the same rules as SOP. And so maybe the sensory components of memories are represented in the dorsal hippocampus, whereas the emotional components are represented in the ventral hippocampus. And I'm just going to give you one very quick set of experiments that might shed some light on why boosting synaptic plasticity in brain regions like the hippocampus might lead to learning changes that could underlie an improvement in mood, which build on these ideas of priming, and in particular the idea that there might be a separate emotional and sensory component to memories. So I don't have any data with SSRIs, unfortunately, but what I'm going to show you is just a couple of experiments from animals that lack the serotonin transporter. So as I'm sure most of you know, the serotonin transporter is the protein that's responsible for sucking up serotonin after it's been released. It's the site of action of SSRIs. These mice are knockouts of the serotonin transporter. So they have no transporter, so 5-HT levels are chronically elevated in these mice. So you could argue that they're a bit like a chronic treatment with an SSRI. The first thing to say about these mice is they're much more anxious than wild type animals. And that's potentially consistent with studies in humans looking at the polymorphism in the promoter region for the serotonin transporter, albeit that's a particularly contentious and controversial story. Okay, so we looked at these mice on a discriminative aversive learning task. So just a basic fear conditioning task where there's a CS plus and a CS minus. So the CS plus is paired with a mild foot shock. The CS minus, there's no shock delivered. And what we found was that the SERT knockouts were much faster to learn this discrimination. So this is conditioning day 1 and conditioning day 2. You can see that the knockouts are doing much better than the wild types. This discrimination index reflects how much more they freeze to the CS plus compared to how they freeze to the CS minus. Okay, and you can see that the knockouts are doing much better than the wild types. Okay, so what exactly are the SERT knockout mice learning? So if we break this discrimination index down in terms of how they respond to the CS plus and the CS minus, we see something very interesting. So actually responding to the CS plus across conditioning days is very similar in the wild types and the knockouts. So the rate at which they learn about the CS plus leading to shock is actually very similar. And where they differ is how they respond to the CS minus. You can see that even on the first day, the knockout animals are moving around much more, so freezing less when the CS minus comes on compared to the wild type animals, suggesting that they might be faster to learn about cues that predict that the bad thing isn't going to happen. And interestingly, we see something similar in extinction. So if we take the CS plus cue and then extinguish it over several sessions, you can see that extinction is much faster in the knockouts than it is in the wild types, okay? And people have argued that extinction and learning about cues that predict that shocks won't happen are examples of inhibitory learning. And we know from SOP and from ESOP that priming promotes inhibitory learning. If you think about something bad, if you think that something bad might happen and then it doesn't happen, then you will learn about the cues that predict that it didn't happen. And so by boosting priming, maybe we can boost inhibitory learning processes that might underlie processes like extinction. Indeed, there are a number of high-profile studies in the literature out there suggesting that SSRIs might facilitate or enhance the speed at which extinction happens. Okay, so just to wrap up then, so hopefully you can see that there are different flavors of synaptic plasticity in the hippocampus, different forms of LTP with different underlying mechanisms. And hopefully what I've tried to convince you today is that maybe these different forms of plasticity might underlie different aspects of priming. In the context of Wagner's model, we've suggested that GluA1-dependent LTP in the hippocampus might be important for self-generated priming, whereas GluA1-independent LTP might support retrieval-generated priming. And of course, these two potentially are additive in terms of their effect on the overall A2 state in the model, potentially reflecting their additive nature at synapses as well. An intriguing possibility is that actually LTD during slow wave sleep may underlie dishabituation, whereby elements of stimulus representations decay back from the A2 state to the I state. By extending these ideas to include sensory and emotional components of memories, then maybe we can start to think about how boosting synaptic plasticity and boosting priming might lead to the sorts of learning changes that might underlie an improvement in mood. In terms of the LTP memory hypothesis, we started off by looking at various issues in the literature that required reevaluation. So how does a priming account deal with these different questions? Well, obviously the time course of learning-induced LTP in the Whitlock study, which was a couple of hours, is much more consistent with a time course that one would expect from priming. In Wagner's models, he suggested that the activity in the A2 state might last for a couple of hours, which is very consistent with what Whitlock sees in terms of their GluA1 and GluA2 levels in the postsynaptic density of neurons. It's also potentially consistent with the idea that whatever memory traces are formed in the hippocampus during waking behavior might be erased during sleep, and actually that slow wave sleep and the depotentiation that happens during slow wave sleep might be an important driver of dishabituation, whereby elements decay from an A2 state back to an inactive state. Multiple mechanisms of LTP that are additive at a single synapse might be explained by the fact that there is self-generated and retrieval-generated priming. And then hopefully you can see from some of the data that I've shown that actually a priming account might fit better than the traditional textbook LTP memory hypothesis. One thing that we haven't really touched on is the idea that plasticity in different neural circuits and different brain regions will do different things, of course. So for example, LTP in the amygdala is going to do something different from LTP in the cortex and potentially different from LTP in the hippocampus. And even LTP in different subfields of the hippocampus. So for example, CA3 and CA1 may play important different roles in different aspects of these behaviors. And we're trying to get our handle on these things by looking at subfield specific knockouts in more detail. And finally, just to say that I think if we think that LTP is an important target for these different psychiatric disorders, then we need to rethink what we think LTP is actually doing. And maybe by targeting specific forms of LTP that might map onto self-generated and retrieval-generated priming, we might do better at developing new treatments for these different psychiatric disorders. Thank you very much for listening. That wasn't too long. Lots of people that I need to thank. In particular, I want to single out two people. David Sanderson, who's now a lecturer up at the University of Durham, who's very much the inspiration for the early work on GluA1 and SOP. And then Marius Panay, who's now a postdoc with Jeff Schoenbaum in the States, who did a lot of the recent studies with the GluA1 knockouts, the voltammetry and the sensory preconditioning, but lots of other people as well. Thank you very much for listening.
Thank you, David. That was an absolutely brilliant talk. I don't know if you want to stop sharing your screen and people can join, just open the videos and.
Are you back?